Today let's discuss the continuation of Pinus. So first we will see the structure of young Pinus stem. So young Pinus stem consists of four parts. The TS of young Pinus stem consists of four parts, that is the epidermis, cortex, stele and the pith. The epidermis is the outermost region, cortex is the next region, stele comes below the cortex, and then the inner region is the pith. Epidermis is the outermost multicellular unilayer. It is a single layered outermost layer. The CS, the cross section of the pinus stem, is irregular in outline due to the presence of scale leaves. The epidermis contains cuticle. It gives protection to the internal tissues. The cortex, it is present below the epidermis, is made up of three regions: the outer hypodermis, the middle parenchyma and the inner endodermis. So the cortex is differentiated into three regions. What are they? Outer hypodermis, middle parenchyma and the inner endodermis. Okay, now let us see what are these three regions. The outermost region of the cortex, that is which is seen below the epidermis, is called the outer hypodermis, and it is made up of 2 to 3 layers of sclerenchyma cells, which this sclerenchyma cells give mechanical support. The middle layer consists of parenchyma cells, which is involved in storage. It also contains resin cells. The resin cells are surrounded by glandular epithelial cells that secrete turpentine. That's why Pinus has its characteristic smell. The inner parenchyma cells arranged in the single layer is called the endodermis. The stele comes after that. It is a conductive tissue consisting of pericycle and vascular tissue. Okay, now the resin canals which are present in the cortex are the diagnostic characters of Pinus stem. Okay, now coming to the stellar region, we said the endodermis is a single layered structure, isn't it? So it is pericycle is seen just below the endodermis. Pericycle is multicellular and it is few layered, and it forms the outer layer of the vascular tissue. Vascular tissue is consist of 5 to 8 bundles. It is separated, each bundle is separated by medullary rays cells. The vascular bundles are arranged in a ring, and each vascular bundle is collateral and open. Open because the cambium is present. Collateral because xylem and phloem are arranged in the same radii. Xylem, it is made up of tracheids and xylem parenchyma. Vessels are absent. The absence of vessels is a characteristic feature of gymnosperms. So the xylem is water conducting tissue, and it consists of tracheids and xylem parenchyma. Vessels are absent. Xylem, two types of xylem can be seen: protoxylem and metaxylem. Protoxylem is the primary xylem and metaxylem is the secondary xylem. Xylem is endark, in which metaxylem is seen towards the outer region and protoxylem is seen towards the center of the stem. Phloem, it is made up of sieve cells, sieve plates and phloem parenchyma. It lacks companion cells. That means companion cells are absent. Pith is the tissue which is present in the center, which is made up of parenchyma cells. Okay, and it is connected with the cortex by medullary rays. So the medullary rays which are present in between the vascular bundles connect the cells of the pith and that of the cortex. Now phloem is arranged towards the periphery and xylem is arranged towards the center of the stem. Okay, now let us see the internal structure of old stem. Now when the stem gets matured, when the pinus stem gets matured, it shows secondary growth. Therefore the structure of primary stem, the secondary stem or the mature stem consists of the outermost region called the bark. It is a dead cork tissue. Okay, It is formed from cork cambium or phallogen towards the outside. The cork cambium or the phallogen is a lateral meristem which gives out secondary tissue towards the outer and the inner side. Now coming to the secondary cortex, which is present just below this bark, it is made up of parenchyma cells. Some cells are filled with tannin. It consists resin canal cells also. Below the secondary cortex, we can see that the vascular tissue is present. The vascular tissue consists of primary phloem, first layer, second layer secondary phloem, third layer secondary xylem, and fourth layer primary xylem. Primary phloem, it is the first form, that is which was present in the primary structure. Due to the secondary growth, this layer gets crushed, and the cambium which is present in between the xylem and phloem is responsible for the secondary growth, and because of production of more phloem and xylem, the primary phloem gets crushed, and it cannot be differentiated in most of the stem. Okay, now the next layer will be secondary phloem. Now the secondary phloem is produced by the presence of or the function of cambial ring, and it consists of sieve tubes and phloem parenchyma, just as the primary phloem. This is also due to the cambial ring, which has the lateral meristematic activity, is responsible for the production of secondary phloem and secondary xylem by forming a continuous ring of vascular bundle. Of cambium. The secondary xylem is also called the wood. It is which is also formed just as the secondary phloem due to the activity of the cambial ring. Okay, now this secondary xylem, as we have already seen, forms the pycnoxylic wood, which is a characteristic of conifers. We've already learned that gymnosperms shows two types of wood, that is the monoxylic wood and the pycnoxylic wood. In Pinus we can see pycnoxylic wood. This secondary xylem consists of tracheids and xylem parenchyma with vessels absent. Okay, and it is traversed by secondary medullary rays also. Now the primary xylem, which was already present in the younger vascular bundles, it is end dark, we have already discussed, and in the central region we can see pith. But when we compare the younger wood with the older wood, we can see that in older wood, pith is the amount of pith will be less when compared to that of the younger stem. Okay, it is made up of small and parenchymatous cells, and it is mainly filled with tannin cells. So this is the structure of old stem. Now coming to the reproduction, the reproduction in Pinus is of sexual type. Okay, here it is written wrongly. It is written asexual reproduction, but the reproduction is by sexual type. We know the plant body is sporophytic, and the Pinus, it reproduces sexually by the production of specialized structures called cones. There are two types of cones, male cone and the female cone. Pinus is unisexual, meaning male and female cones are separate. It is heterosporous, meaning it produces two types of spores, that is microspores and megaspores. Microspores are produced in the male gametophyte and megaspores in the female gametophyte. Pinus is monoecious. That means they are produced separately on separate branches. The male and female cones are produced on separate branches. Now, when we take the male cone, it is also called staminate strobilus. These male cones are formed in clusters in the axial of scale leaves at the tip of the long shoot. Okay, each cone has a central cone axis, and around the cone axis we can see numerous microsporophylls which are arranged spirally. Each microsporophyll has a small stalk with a terminal leafy expansion. It expands like a leaf, and each microsporophyll produces two microsporangia on its lower side. In the microsporangia, it consists of two wall layers, archisporial cells and microsporangial wall. So, male's reproductive organ or the staminate strobili are produced in clusters. So, what we see here is a cluster of male cone. That is what we are seeing here. Each grape-like structure is the male cone. So, each male cone, when we take, we can see the cross section of it. Each male cone consists of microsporophylls. We can see leaf-like microsporophylls. They are arranged spirally on the cone axis. When you take one microsporophyll, we can see that the microsporophyll consists of a tiny stalk and two microsporangia. Okay. Now, the inner archisporial cells, the inner cells, they divide to form microspore mother cells or pollen mother cells, and the outer wall layer is called the microsporangial wall layer, which is multi-layered, and there is an inner layer called the tapetum which gives nourishment to the microspore mother cell. Okay, each microspore mother cell, it undergoes meiosis, produces four haploid winged pollen grains. So the pollen grain out here is winged, it has wings, and this winged pollen grains are called microspores. These pollen grains develop into the male gametophyte. So what are the features of the microspore or the microsporangia? The microsporangia consists of wall layer which is called the microsporangial wall, which has an inner wall layer called the inner structure called the microsporangia mother cells, which divide to form the microspores. This is the structure of a microspore. We've seen that it is winged. No, so the microspores are small and it is winged. Okay. Now coming to the microspore, each microspore or the pollen grain, if you see, it has two wall layers called the outer exine and the inner endine. The inner protoplasm, it contains more cytoplasm and haploid nucleus. The outer exine is rigid, it is made up of cutin, and it expands, that is the outer exine expands to form the wings. And this is the structure of the microspore. Now the inner indine, it is elastic, okay, and it breaks open to form the pollen tube, which is formed during pollen germination. Because they are winged, these pollen grains are dispersed by wind. Now what we see here is the female cone of Pinus. Okay, now when the female cones, when they are younger stage, they are green and fleshy, but when it becomes mature, it becomes brown and woody. The female reproductive organ, also called the ovulate strobilus, are formed in the axil of scale leaves at the base of the long shoot, where it is formed at the base of the long shoot. Each cone, as we have seen, has a central cone axis on which numerous megasporophylls are arranged spirally. Each megasporophyll consists of two kinds of scale leaves called the bracts scale and the ovuliferous scale. Okay, now we can see that the female cones are also found in clusters, but the number of female cone in one cluster is lesser. It may be 4 or 5 female cone in one cluster, but in microsporophylls we've seen that the number of the male cone will be 50 to 100 in one cluster. Okay, now this is the cross section of the female cone. We can see the ovuliferous scale and the bract scale. Now the bract scale is small, it is thin, membranous, they develop from the cone axis. The ovuliferous scale is stout, It is thick and woody. It develops from the base of the upper surface of the bracts scale. The ovuliferous scale produces two megasporangia or ovules at the base on its upper surface. The megasporangium produces only one functional megaspore, and that develops into the ovule. Okay, so that is the difference. Now when you take microsporangia, it produces many microspores, whereas in megasporangium it produces only one functional megaspore. That's the difference. Now when we take the structure of ovule, each megasporangium or ovule, it consists of two parts. What are they? Integument and the nucellus. The outer integument, it is made up of three layers. What are the three layers? The outer fleshy layer, the middle stony layer, and the inner fleshy layer. Okay, the inner nucellus is the nutritive layer. So the wall layers are formed by the integument, and nucellus forms the nutritive tissue. Now let us see the cross section of what we have studied, the structure of ovule. So this is the cross section. We can see the blue layer is the middle stony layer. We can see the outer and inner fleshy integuments, and the pink colored region is the nucellus, which provides nourishment for the inner blue colored region, which is the which forms the megaspore cells. The integument leaves an opening called the micropyle. During the development of the ovule, a cell in nucellus near to the micropyle is differentiated, and it acts as the megaspore mother cell. The megaspore mother cell undergoes meiosis to form four haploid megaspore in linear tetrads. Okay, the three megaspores disintegrate and only one remain functional. Out of four, three disintegrate and only one remain functional, and this functional megaspore, it develops into the female gametophyte. Okay, so this is the structure. Let us just rewind once again. The integument leaves an opening called the micropyle. It is not fully covered. It leaves an opening called the micropyle, and the nucellar tissue, which is seen just below the micropyle, it forms the megaspore mother cell. This undergoes meiosis to form four haploid megaspores, out of which three disintegrate and only one remain functional. This functional megaspore develop into the female gametophyte. Hope you understood. Thank you.